All right, here is our second video for genetics, Intro to Punnett square word problems. So we are going to go ahead and look at some example genetic crosses. So we're going to start off with a heterozygous tall pea plant is crossed to another heterozygous tall plant. Tall is dominant to short. And then we're going to have to calculate the phenotype and the genotype ratios of the offspring. So which trait or allele is dominant? We know that it's tall, and we need to assign a letter to go with that, so we're going to use the capital T. And then the trait or allele that is recessive is short, and we'll use the lowercase t. So the next thing that we need to go ahead and do is we need to think about both parents. So both parents are tall, as indicated in our problem up here. We know that both parents are heterozygous, which means that they have one copy of the tall gene and one copy of the short gene. So capital T, lowercase t, capital T, lowercase t. And when both pea plants go through the process of meiosis, we know that they can go ahead and they could either give the allele capital T or the allele lowercase t. All right. So now that we have that taken care of and we have figured out what our word problem is actually talking about, we need to go ahead and we need to do a cross. So in doing a cross, you separate the alleles away from each other, which is the same as the process of meiosis 1 into meiosis 2. It's just a different visualization for it. This is the end product of what would be in the sperm or be in the egg. And much like when we did our pop beads lab, we're now going to go ahead and move our alleles into the boxes. And so right now, this box here, this is haploid, and this box here is haploid, but now this box here is diploid, and this is capable of becoming a new pea plant. So let's follow suit and fill in the rest of our boxes. So now the next thing that we are asked to do is write the genotype ratio in the offspring by counting how many of each genotype are in the Punnett squares offspring boxes. And our offspring boxes are 1, 2, 3, 4. So the genotype ratio is 1, capital T, 2, capital T, lowercase t, and 1, lowercase t, lowercase t. The phenotypic ratio, or how the plant looks, is can be brought on by looking at the genotypic ratio. So we know that this one is going to go ahead and be tall. This one will be tall. This one will be tall. This one will be short. So we have 3 tall to 1 short pea plant. So Punnett squares are named after the man who invented them. They are a tool that can help scientists figure out 2 things: all the possible combinations of the alleles an offspring have. They can also figure out the probability of each allele combination in the offspring. So let's go ahead and we're going to try out some word problems now by using some different clues based upon different organisms. So we have a young striped zebrafish, had a striped father and a spotted mother. So the first thing I want to do is underline, where are we getting the genes from? And we know stripes is dominant over spots. So the first question is what is the genotype of the young striped zebrafish. So I think we need a little bit more information before we can go ahead and figure out the answer to that question. So because the young fish has stripes and its mother was homozygous for the recessive trait spots, you can tell that the young fish is a heterozygous. The mom would have only been able to give lowercase s or lowercase s. All right, so it must have inherited one recessive, and in this case, lowercase s allele from its mother. So if we wanted to go ahead and make a really quick Punnett square right here, all right, I'm gonna put mom along the side and I'm gonna give lowercase s and lowercase s. We know that dad had stripes, and so he's a capital S. We don't know if dad was heterozygous or homozygous for stripes, but we do know that he had the dominant gene, so we don't have anything to put here. But knowing that information, capital S, lowercase s, and capital S, lowercase s, and knowing that our young zebrafish is striped, that must mean that our zebrafish is capital S, lowercase s, and is a heterozygous. All right, on to the next one. So oftentimes we use this word hybrid. Hybrid simply means heterozygous. We could have said that our striped zebrafish was a hybrid. In this example, because the heterozygous shows the dominant trait, brown hair, that is dominant. So, hybrid. An albino male mouse is mated with a hybrid brown-haired female mouse. What is the genotype of the female parent? So, a brown-haired female mouse. So that must, and it's a hybrid. So we can say capital B for brown, lowercase b for albino. Albino, they would be white. All right, so our male mouse would be lowercase b, lowercase b. And the question asks, what is the genotype of the female parent? Capital B, lowercase b. So while we use this term hybrid to mean heterozygous, all right, we can use another term, true breeding, to mean homozygous. So they have two of the same alleles. So a true breeding brown-bodied male fly mates with a white-bodied female fly. The brown body allele is dominant over the white body allele. We're going to use B for brown and lowercase b for white. What is the genotype of the male parent fly? Well, we know that it is a true breeding brown, and the problem has told us to use B for brown, and we know that true breeding means homozygous. So the male would be capital B, capital B. All right. Lastly, dominant or recessive. So a heterozygous round seed pea plant is crossed with a wrinkled seeded pea plant. The question is, which allele is dominant? So to figure out which allele is dominant, we need to look at the phenotype of the heterozygous individual in the problem. And the heterozygous individual has the dominant trait, which is round seeds. You can see all of them are round here. Okay. All right. Moving on. So, goldfish. So, in goldfish, there is a trait caused by a dominant allele in a developmental gene called bubble eye. Bubble-eyed goldfish have a large bubble-like sac near each eye, and they lack a dorsal fin. So see how there's no dorsal fin up here, and they have this bubble here. Normal eyes is categorized by lowercase b and is recessive. So the only way a goldfish can have the normal eye situation is if they were lowercase b, lowercase b. So let's look at our two goldfish here. A normal-eyed fish genotype must be lowercase b, because normal eyes is recessive. Notice that even though normal eyes is recessive, it's going to be more common actually in the population. All right, the dorsal fin is going to help them swim, and it would be good to not have a bubble-like sac near your eye for vision purposes. The bubble-eyed goldfish's genotype could be capital B, or capital B, lowercase b. There's no way of telling by just looking at it. So one of the ways that we would go ahead, and one of the ways that Gregor Mendel went ahead and he started to determine which traits were dominant and which traits were recessive, was he did something called test crosses. So the question is how can you figure out the bubble-eyed goldfish's genotype? Do you know, are they capital B, or capital B, lowercase b? So scientists do test crosses to see if an individual is heterozygous or homozygous dominant for a particular trait. They do this by crossing the individual with the unknown genotype with a homozygous recessive individual, and in this case, it's lowercase b, lowercase b. So when we go ahead and we look at our offspring here, we know that all of these offspring without the bubble eyes are lowercase b, lowercase b. And we know that the ones that have a bubble eye are capital B something, capital B something. When we look at the parents, we know that this parent must be lowercase b, lowercase b. And this parent, based upon the offspring that are here, must be capital B, capital B. Sorry, my grandfather clock is going. All right, so the parents, grandfather clock messed me up. I'm so sorry, guys. This parent is lowercase b, and then this parent, to end up with some lowercase b offspring, has to be capital B, lowercase b, all right? And then the other parent is lowercase b, lowercase b. To go ahead and have all bubble-eyed offspring, that means that this parent needs to be capital B, capital B. So we can go ahead and we can perform a cross really quickly. Well, two actually. Here's our lowercase b, lowercase b. So this is the same for both crosses. And then to end up with version number 1, capital B, lowercase b. All right, so this is our cross to show what happened there. And then to show what's happening in this one, I'm going to do the Punnett square up top here. Lowercase b, capital B, capital B. And then all of these will be bubble-eyed goldfish. All right. Those are your quick notes on how to go ahead and do Punnett squares and differentiate between different word problems.